tell you that obesity is growing all over the world, not in Kuwait only. And we know that overnutrition and high-fat diet actually leads to obesity, and obesity actually induces inflammation, and that inflammation actually results in insulin resistance and type 2 diabetes. If you look at this cartoon, you can see that, I don't know if it's going to work. It's hard to work. Okay. And there's obese adipose tissue. As you can see, the size of the adipocytes is bigger, and the number of infiltrated macrophages or immune cells are more in case of obese adipose tissue as compared to lean adipose tissue. You see, the number of immune cells are less in this adipose tissue, and also the size of the adipocytes is also less as compared to the obese individuals. So these infiltrated immune cells actually communicate with the adipocyte and then trigger inflammation. In my next slide, if you look at it, there is a crosstalk between adipocyte and macrophages. Adipocytes produce a lot of pro-inflammatory cytokines, chemokines, and these pro-inflammatory cytokines, chemokines actually bind to their respective receptors on the infiltrated monocyte or macrophages, and then they activate pro-inflammatory genes and then produce a lot of inflammatory, again, cytokines, particularly TNF-alpha and IL-6. And then this time, TNF-alpha, IL-6 again, attack the adipocyte, and then adipocyte produce more cytokines or chemokines. In this way, actually, the inflammation actually came up in autocrine and paracrine fashion. So eventually, this leads to insulin resistance. And one thing more I would like to highlight in my talk also, the free fatty acid. In case of obesity and diabetes, there are more free fatty acid in the circulation, and they also play a significant role in inducing inflammation. This is a classical pathway in which like insulin play a role. So insulin bind to insulin receptors, and then there's an autophosphorylation occurs in some proteins, and then those proteins, activate the glucose transporter protein to the plasma membrane, and then glucose uptake is permitted. And by this way, glucose, lipid, and protein metabolism regulate in really nice coordinating way. So this slide actually shows that how pro-inflammatory or inflammatory mediators play a role in insulin resistance. As we know that TNF-alpha and IL-6, these are the signature of inflammation and insulin resistance. So their production is high in case of obesity in obese individuals and type 2 diabetes. So TNF-alpha and IL-6 actually, they induce the phosphorylation at the serine site of insulin receptor substrate. And this insulin, when this site gets phosphorylated, so the AKT pathway activation gets blocked, and then the glucose transportation also stops. So similarly, if you go different free fatty acid or fat metabolites also play a role in the insulin signaling pathway. So they also block AKT phosphorylation and insulin receptor tyrosine phosphorylation, and then induces insulin resistance. So in most of the cases, if you look at this, you can see that NF-kappa B playing a significant role in terms of production of the inflammatory cytokines, chemokines, and other inflammatory mediators. So today, actually, I will cover up the following areas. So I will give a brief talk about free fatty acid, role of free fatty acid, fat metabolites in insulin resistance. And then I will give a talk about the role of immune receptors, TAR-like receptors, in causing insulin resistance. And then I will also talk about transcription factors, NF-kappa IRF3 in obesity and insulin resistance. And finally, I will give a brief talk about the role of epigenetics in insulin resistance. So as I said that because fat metabolism get disturbed in case of obesity and type 2 diabetes, so then the free fatty acid in circulation are very high in case of obesity and type 2 diabetes. And we also recently found that if you look at that slide, and you can see that the expression of, there's an increase in circulation of free fatty acid in case of obese individuals, along with the other endogenous, the markers. So now, so as I said that free fatty acid or fat metabolites are commonly, the acetyl-CoA, diacylglycerol, and ceramide. So ceramide is very prominent, and it has been found that ceramide actually activate NF-kappa B and also induces a lot of inflammatory mediators in immune system. So recently, actually, a study has been done, and it's published in Cell Metabolism. And one group, they found that when there is a deficiency in ceramide, so it prevents from the insulin resistance, and it prevents the individual from the insulin resistance. So they use actually animal model, and they block actually the production of ceramide in those animal models by knocking out some ceramide synthesis enzyme. So they found that if you look at the figure E subfigure, so that there's those mice who are ceramide deficient, actually they gain less body weight as compared to the wild type. And if in figure F, you can see they have less body fat as compared to the wild type mice. And in figure I, you can see that they also reduce, ceramide deficiency also affect the inflammatory mediators. So those mice who are negative, they are knockout of ceramide, so they have less inflammatory mediators as compared to the wild type, as you can see in this figure I actually. And then next, this group also found that what is the impact of those inflammatory mediators on the energy expenditure. And then they found that those mice actually having, show high energy expenditure as compared to the wild type. And finally, they found that those mice show glucose tolerance as compared to the wild type mice. So it means ceramide play a significant role in insulin resistance. And if you look at this cartoon, you can see that obesity, and on the right side is diet-induced obesity. So when we knock out this ceramide synthesis enzyme, so these mice improved glucose tolerance. So this shows that ceramide is a key regulator in inflammation and insulin resistance. So next, since this slide, I just give a brief, the molecular mechanism of another two metabolite, acetyl-CoA and diacylglycerol, because their concentration also high in case of obese and diabetic, type 2 diabetes individuals. So if you look at the first cartoon, you can see that when acetyl-CoA is increased in case of type 2 diabetes or obesity, because there are a lot of lipolysis, and it produces also IL-6, and also increase the production of glucose, so as compared to the case of the normal individuals. So acetyl is also involved in the production of glucose in liver. So in the right side, other side, how the other metabolite affect the insulin resistance. Diacylglycerol actually phosphorylate protein kinase C, and then protein kinase C actually activate the serine phosphorylation on the insulin receptor substrate, and then they induce insulin resistance through the AKT cascade. So I just say that this shows that, because as I said, not much work has been done related to fat and fatty acid metabolites in terms of insulin resistance. So the work I presented is recently has been done, and they figure out that fat and fatty acid metabolites play a significant role in the induction of insulin resistance. So next, actually free fatty acid is also the ligand for the TAR-like receptors. So that's why most of the work is actually focused on TAR-like receptors or immune receptors in terms of induction of insulin resistance. So TAR-like receptor basically a pattern recognition receptor. They are highly expressed on immune cells. They are divided into two groups. One is plasma membrane receptors, another one endosomal receptors. And these receptors, bind with different pathogens as well as different ligands, particularly cell-degrading products as well. And then they get activated, and then they activate NF-kappa B transcription factors. And they also, some TAR-like receptor, particularly TLR4, actually it works through two pathways. One is dependent on NF-kappa B, other is interferon regulatory factor 3. So based on this information, and as I know that TAR-like receptor or pattern recognition receptor play a significant role in immunological or inflammatory disorders, so we hypothesize that change in TAR-like receptors 2 and 4 expression and TLR signaling in obesity may mediate induction of insulin resistance. So to test this hypothesis, actually, we collected the blood sample, PBMC, from obese and lean individuals with and without diabetes. And we also collected subcutaneous adipose tissue specimens. So we found that if you look at this figure, I will try. In the first, there is a significant increase in the expression of it's not working. Okay. So that there is a high expression of these TAR-like receptor 2 and 4 in obese individuals, particularly in PBMC, and then we also tested the expression of these TAR-like receptors, particularly in adipose tissue. As we know, that adipose tissue is endocrine compartment in inflammation. And you can see that in adipose tissue, particularly, there is a lot of infiltration of the monocytes or macrophages, which show the expression of these TLR receptor, TLR2, TLR4, and this receptor also correlate with the BMI. So it means obesity triggered the expression of those receptors in obese individuals. Next, we also looked at if the TAR-like receptor expression is high, what about the signaling molecules which TAR-like receptor use to activate inflammatory genes? So we also tested and we found that the adapter proteins like IRAK, MYD88, they are also highly expressed in PBMC and also highly expressed in case of adipose tissue. And next, we want to see whether in these subjects, that the inflammatory signature TNF-alpha and IL-6 also expressed or elevated. So we found that there is a significant expression of, sorry, that is time. Okay, I will just skip the few slides and then I go down. So we found that TAR-like receptors are enhanced and adapter proteins along with the cytokine enhanced in obese individuals, and they lead to insulin resistance. Next, we look at the other, important marker, which is interferon regulatory factor 3. And this work actually we have done in collaboration with the Harvard School of Medicine, and we found in global knockout mice that the expression of IRF3 is reduced as compared to wild-type mice. So that interferon regulatory factor play a role in the modulation of inflammatory mediators. And then next we test how it affects on the metabolic marker or parameters. So we looked at the body composition and food intake. Although these mice, who are IRF3 deficient, they take more food as compared to the wild type, but they have less fat and high lean mass. And so it means IRF3 also positive regulator of adiposity. Next we look at what is the energy thermogenesis in those mice. How it was. So we measured this thermogenesis or energy expenditure in terms of oxygen consumption, carbon dioxide production, heat production, and movement. So however the mice have having the same movement, same mobility, but they use the IRF deficient mice, they use more oxygen and produces more carbon dioxide and heat production. So it means IRF3 play a significant role in energy expenditure. So based on this one, we also tested maybe this IRF3 play a role in the regulation of regulation of like a thermogenic and mitochondrial gene, and we also found that this IRF also regulate these mitochondrial and thermogenic gene in those mice. So and also the mice which are deficient in IRF3, they show increased glucose tolerance as compared to the wild type mice. And so I don't have time for this. Okay, so we recently found that different nutrients, just I give the brief, we found that different nutrients have different impact on the chromatin modification. So we have actually a good amount of data, and this work actually we are doing in collaboration with Stony Brook University. And hopefully in coming year, I will present the conclusive the information related to my this unpublished data. So at the end, I would like to thanks to Kuwait Foundation for the advancement of sciences, for their for providing us funds, and thank you very much.
Thank you, Dr. Rashid. As I told you, molecular biology is something very tough for us to understand. But this is just to show you how important is, bench research. And bench research usually creates ideas we're actually going to translate for us as clinicians later. So just to give you a glimpse of what's happening down in the molecular level. So any question? Any immediate question? Okay. Did you understand anything? That's good. At least we know that there is something going on, and there are lots of markers that actually people are working on, and here will start the medicine in the future. Thank you, Dr. Rashid. We'll give you your... Thank you very much. Our last speaker for the session, the first session, Dr. Hassa Al-Kandari. Dr. Hassa Al-Kandari, she's a colleague. We worked together for many years. We studied together in university, in Kuwait University. Dr. Hassa is the chairman of Diabetes Endocrine Pediatric in Farwania Hospital. She had the Saudi Board in Endocrine and Diabetes, and she is the fire in Kuwait Diabetes Society, and we work together also in the Ministry of Health and in the pump committee, where we're actually studying the pumps and the sensors, and she's one of the most knowledgeable pediatricians in this field. Thank you. So she's going to talk to us about something very interesting, which is monogenic diabetes. And we're going to hear from her.
Assalamu alaikum. Bismillah ar-Rahman ar-Rahim. Maybe you're tired after 3 series of lectures and the last one with a huge dose of knowledge. Mine will be very simple, inshallah. It will be very short and more clinically oriented. Today we'll speak about monogenic diabetes. I'll start with a few clinical cases just to keep you excited for the lecture. First, we have Noura, who's a 30-year-old girl, a female, was diagnosed to have diabetes since the age of 16 years. She had positive C-peptide and strong family history of type 2 diabetes in her mother and sister. She always required small dose of insulin to maintain good glycemic control. So how many of you thinks that Noura has type 1 diabetes? It's either type 1 or type 2. She was labeled as type 1 diabetes. We'll move to the second case. Baddah, 13-year-old boy, diagnosed with chronic renal failure soon after birth due to cystic renal disease. He had renal transplantation at the age of 3 years. And at the age of 9, he started to show some intermittent symptomatic hyperglycemia with the use of steroid and with infections. And then at the age of 10 years, his hyperglycemia was persistent, and he was labeled as diabetic, and he required insulin therapy. Baddah has no family history of diabetes. So again, Baddah was labeled as type 1 diabetes. Then coming to third case, which is Razan, 13-year-old Lebanese girl, eldest of a girl of a consanguineous parents, also had diabetes since the age of 10 years, but she had severe acanthosis nigricans, coarse features. Her younger brother, who's 2-year-old, had similar features but no diabetes, but also with high insulin level. So the 3 cases had diabetes. It's diabetes, but what type? And does it matter to know the type of diabetes in the example of cases that we've shown before? We have 2 common types of diabetes. It's 4 types, but the 2 common types that we see in the clinics are either type 1, which is when beta cell function is affected and you have deficiency in insulin, and these patients are insulin dependent, and they do usually have antibodies as it's an autoimmune disease. And then type 2, when the person is normally thought to have type 2, he doesn't have type 1 or other types of diabetes. He might be obese and have acanthosis nigricans. So our patient used to be either type 1 or type 2. When we were taught in medical school, and even when we start our clinical practice, children presenting with polyuria, polydipsia, and high blood sugar are type 1 diabetes, and we rarely thought of other type of diabetes in children. So we did not consider them, we did not look for them, even you start insulin without doing the basic workup. So when shall we suspect that type 1 diabetes in children is not correct? If diabetes developed before the age of 6 months, there is 1% chance that he's still having type 1 diabetes. And if there is family history, one parent is having diabetes of similar degree. And if there is still evidence of endogenous glucose production after 3 years of diagnosis, extended honeymoons after 3 years, you can detect C-peptide level in the blood. And when you have absence of antibodies at diagnosis, especially at diagnosis. Coming to type 2, when shall we suspect that type 2 diabetes might not be correct? When there is no markedly obesity, or there is a diabetic member in the family who have normal weight too. The chance is only 20% that it still be type 2 diabetes. And when there is no acanthosis nigricans, and when the patient comes from an ethnic background that do not have high risk of diabetes or have tendency of diabetes, and when you don't have evidence of insulin resistance, that's the C-peptide is within the normal range, not high. If you have these features, you need to suspect. Sometimes you need to reassess your patient. Sometimes you need to do more workup to reach the proper diagnosis of your patients. So today we'll be talking about monogenic diabetes, what used to be called before maturity onset diabetes of the young. It's a new disease, or it's a new diagnostic type of diabetes, first described in 1964. And it was applied to any child or young adult who had persistent asymptomatic hyperglycemia. So it was mild, without progression to acidosis. So the feature was mild diabetes, young adolescent or adult, and no ketoacidosis. They used to label it as hereditary form of diabetes because they've noticed that it passes through generations, and it was taking a Mendelian type of inheritance, so they presumed that it's autosomal dominant. And if it is autosomal dominant, then it's most likely it is monogenic form of diabetes. And the name now is monogenic diabetes. The definition is that it's an inherited form of diabetes when you have single mutation or more than one mutation in a single gene. It has to be a single gene. It may be dominantly inherited, recessively inherited, non-Mendelian inheritance. Usually, it's either affecting the beta cell function, where there is reduced production of insulin, or insulin function, where there is resistance. In children, most of the cases come from reduction in beta cell function. We rarely see cases of resistance in monogenic diabetes in our practice. It's grouped into 3 groups: neonatal diabetes. I will not talk about it in this lecture. Familial diabetes, which is the one that is usually we label as MODY, and genetic syndrome, where the diabetes is associated with other features, and they can be all grouped together into syndrome. Many genes have been identified. More than 40 genes defects have been identified, each having a certain phenotype. But there are 3 common genes that are responsible for around 70% of monogenic diabetes. These are type 1, 2, and 3. In type 1, it's due to hepatocyte nuclear factor 4 alpha. And type 2, it's the enzyme glucokinase. And type